Welcome dear students. In this video we are going to learn about a very important cell signaling that is called receptor tyrosine kinase pathway. In my previous video I have mentioned the functioning of JAK-STAT pathway, and in that class I have mentioned that the JAK-STAT pathway and the receptor tyrosine kinase pathway works very similarly, but there is a difference. The single difference is here, in case of JAK-STAT pathway the receptor don't have any intrinsic domain of kinase domain. But in case of RTK pathway, the receptor has intrinsic kinase domain. That is the difference. So at first I will show you what are the ligands that helps in the activation of RTK pathway or receptor tyrosine kinase pathway. This is the main important thing, ligand. Like in case of JAK-STAT pathway I have mentioned the ligands like cytokines. It could be interleukin or interferon, or some hormones like growth hormone, prolactin, thrombopoietin, erythropoietin. These actually activate the JAK-STAT pathway. But in case of RTK pathway, the ligands are growth factor. Many growth factors that are present in the serum are actually helps in the activation of RTK pathway. And a hormone, protein hormone specifically I want to mention here, insulin, can activate this RTK pathway. Okay, the growth factors, the name of these growth factors are EGF. That means epidermal growth factor, NGF, nerve growth factor, FGF, fibroblast growth factor, PDGF, platelet derived growth factor, IGF-1, that is insulin like growth factor 1. Another you can remember MCSF, that means monocyte colony stimulating factor, KIT, a stem cell factor, and ephrin. This is very important thing I will mention in my separate class. Ephrin is a juxtacrine actually molecules that helps in the migration of nerve cells. Ephrin. So in this class we will actually learn about this growth factor protein. They are actually very important in RTK pathway. So in all these cases, in case of RTK pathway, the receptor, let's suppose this is the plasma membrane. In case of RTK pathway, most of the receptor, this is the extracellular domain, this is the transmembrane domain, cytosolic domain. Most of the cases the receptor remains monomeric in nature, and they have a receptor tyrosine kinase. So this is the kinase domain. They have already a kinase domain but inactive in conformation when there is no ligand and they are monomer. But when any growth factor, it could be EGF, NGF, FGF, PDGF, IGF, any growth factor, or VEGF, yes, vascular endothelial growth factor, any growth factor bind with this monomer, this monomeric receptor then dimerize with another receptor. So they dimerize. This actually occurs when ligand binds. So let's suppose this is the another receptor of the growth factor. This is the growth factor. When this dimerization occurs, there is a loop present in this kinase domain that actually prevent the activity of the kinase enzyme. So there is a loop called activation loop, and they actually mask or hide the kinase activity domain of this kinase enzyme. So this loop is called activation loop. This loop actually prevents the functioning of this kinase domain. Now, when these two receptors dimerize, they phosphorylate each other. This enzyme phosphorylate this activation loop, and this enzyme phosphorylate this activation loop. And when activation loop gets phosphorylated, there is a conformational change occur in the activation loop so that it no longer can inhibit the kinase domain and the kinase domain gets activated. When the kinase domain gets activated, they phosphorylate the C domain of the receptor. Phosphorylate. They phosphorylate the C domain of the receptor. Now this phosphate can be the a region where many signaling proteins can bind. These are called adapter proteins. Now I am giving a simple example like PDGF. Scientists have found that in case of platelet derived growth factor receptor, many proteins can bind with this phosphotyrosine, because this is the phosphotyrosine. You have to remember, in case of RTK pathway, that means receptor tyrosine kinase, this kinase domain is specifically target the tyrosine amino acid. All the phosphate I have written here that are the tyrosine amino acid. In also the activation loop have a particular tyrosine that is actually gets phosphorylated. Now this phosphotyrosine, these are the phosphotyrosine, can be targeted by many signaling cells, signaling proteins. One example is PLC gamma. They can attach with these phosphotyrosines. Another is called PI3 kinase enzyme. This is also a kinase that can associate with this phosphate group. There is another protein called GRB2, that is a adapter protein. SIR protein can bind with this phosphotyrosine. SIR protein also a kinase protein, and RAS gap protein can bind with this phosphotyrosine. So many proteins can bind with this phosphotyrosine domain. So you have to understand first thing that this growth factor actually helps in the dimerization of the receptor. Then each of these kinase domain phosphorylate the activation loop. Activation loop actually prevents the activation of the kinase activity. Then when activation loop gets phosphorylated, they no longer actually mask or hide the kinase domain. Then kinase domain actually phosphorylates the C-terminal domain of this receptor. And when this gets phosphorylated, the C-terminal domain of the receptor gets phosphorylated, different proteins bind with this phosphorylated phosphotyrosine. Now PI3 gamma can activate calcium level, increase the calcium level, and that actually helps in many different cellular functions. PI3K can activate a kinase called PKB, protein kinase B, also known as AKT pathway. AKT, that actually helps in the activation of a pathway called mTOR pathway. You can see here, this mTOR pathway can be activated by RTK pathway also. Another example is GRB2. GRB2 can activate a RAS MAP kinase pathway. So this RTK pathway can also activate RAS MAP kinase pathway. So you can see here, SIR can phosphorylate each other, and RAS gap actually helps in the deactivation of the RAS protein. So one single ligand can activate different cellular response or cellular signaling pathway. Okay. Now here I want to give you a special example of a growth factor receptor that actually works very differently in case of receptor tyrosine kinase, because I want to mention here that this receptor tyrosine kinase pathway, the one important thing is autophosphorylation. This is the first thing, autophosphorylations. That means what I have mentioned here, this receptor phosphorylate this activation domain, activation loop, and this kinase domain actually phosphorylate the this site. So this is called autophosphorylations. These two domains autophosphorylate each other. But in case of EGF, these do not occur. Epidermal growth factor, this is not occur. But how this receptor actually function, this is very important. In case of EGF, the thing is different. Here one receptor has the kinase domain, another receptor has the kinase domain. Okay, they have kinase domain, and when any ligand binds, let's suppose these ligands binds, they actually help in the dimerization of the two receptors, same like this PDGF or any other growth factor. But I have mentioned that in case of RTK pathway, autophosphorylation occur. That means two domain phosphorylate each other, cross phosphorylate each other. But in case of EGF, this is not occur. In case of EGF, what happens, this one kinase actually attach with the other kinase, and they actually helps in the change in the conformation of the activation loop. So the activation loop here is not phosphorylated, but when a kinase domain of a receptor binds with another kinase domain of the receptor, the activation loop gets changed its structure. In case of LODIs, it is said that this domain called donor and this domain is called acceptor. But in Bruce Albert, this same donor is actually denoted as activator, and this acceptor is called receiver. So the name differ in these two books. In Lodis, donor, acceptor; in Bruce Albert, activator and receiver. But the function is same. Here two domain interact, and the activation loop of the acceptor or the receiver actually change so that it becomes activated, but not autophosphorylation do not occur. Then this kinase domain gets activated, and this receiver or acceptor kinase domain phosphorylate each other. They also phosphorylate the C domain of this same receptor and also phosphorylate the C domain of the other receptor. Here I want to mention some few facts about the EGF growth factor, and before I start EGF growth factor, I want to give another example of another growth factor called FGF. In case of FGF3 receptor, it has been found that if there is any mutations by which this FGF3 receptor get actually activated, constitutively get activated without presence of FGF, then it activates its proteins called STAT1 transcription factor. This actually occurs prematurely, and this lead to a disease called thanatophoric dysplasia. Thanatophoric dysplasia. Dysplasia. Okay. What happens? FGF3 receptor, if there is any mutation in this receptor gets activated without absence of FGF. In the blood or the serum, maybe there is no FGF, but if there is any mutation in this FGF3 receptor, then the receptor is always active. Then it activates the STAT1, and then the STAT1 actually block the growth of chondrocyte, so that the newborn baby have short leg, short hands, and also the thoracic rib, also thoracic cage also very short, and they die eventually. This is called thanatophoric dysplasia. So you can see the importance of this RTK pathway. Okay, now I want to give you an example of this EGF receptor very details. In case of EGF receptor, this EGF receptor are named as ERB B1, B2, means ERB B1, ERB B2, ERB B3 and ERB B4. So there are 4 receptors that can bind with EGF. Now in human, the name differs. The name in case of human called HAR1, HAR2, HAR3 and HAR4. HAR means human epidermal receptor. Okay. All these receptor bind different ligands. I want to give you details for your CSIR preparations. The HAR1, this HAR1 receptor binds with EGF, that is normal. Then it can bind with TGF alpha, transforming growth factor alpha, heparin binding EGF, amphiregulin. These are the ligand. Epiregulin, epigen and beta cellulin. So these are the ligand for HAR1 receptor. There is no ligand for HAR2. You have to remember this. There is no ligand for HAR2. The ligand for HAR3 are neuregulin 1 and neuregulin 2. In case of HAR4, the ligands are neuregulin, or I can write neuregulin 1, 2, 3 and 4. Now the HAR1 binds with homodimer. they can bind with HAR1, HAR1 or heterodimer HAR1, HAR2. I have mentioned HAR2 do not bind with any other, but HAR2 actually make heterodimer. HAR2 can bind with HAR1 like here, or HAR3 or HAR4. It make the heterodimer. HAR3 do not bind with HAR3, it bind with always HAR2. And HAR4 can bind with HAR4, that make the homodimer. So two make a homodimer, HAR1 and HAR4. And HAR4 can also bind with HAR2. So there is a very significant role of HAR2, and the structure of this HAR2 is very different from all these HAR1, HAR3 and HAR4. HAR3 do not function itself, it actually associates with HAR2. The HAR2 looks similar when a HAR1 binds with a EGF. Let's assume this is the HAR1. When it binds with the EGF, let's suppose this is the EGF, epidermal growth factor. This is the HAR1 when it is bind with the ligand. But the shape of the HAR2 is very similar like this HAR1. It do not bind with any EGF, but looks like that it is bind with an EGF. Looks similar like it is bind with EGF. But in case of when there is no other molecule bind with this HAR2, it cannot activate this pathway. Okay, so in case of EGF, there is a difference in the activations. Like I have mentioned, the EGF growth factor do not activate autophosphorylation. There is a structural change occur. And another is the receptor. Now the HAR2 is very important for a disease condition called cancer, breast cancer. The HAR2 protein, when there is a mutation occur, specifically a point mutation that changes the amino acid, single amino acid like valine to glutamine, then this HAR2 protein becomes constitutively active. It becomes always active. This is called new protein, new, and new means neuroblastoma, a kind of tumor. Scientists have found neuroblastoma, but it also actually occurs in it founds in HAR2 overexpression, found or constitutively activated HAR2 present in breast cancer. This new protein is called oncoprotein because it actually promote cancer, new for neuroblastoma. And there are two medicines that actually target this new oncoprotein, that is called trastuzumab. This is actually a monoclonal antibody. It is also known as Herceptin, very famous antibody, Herceptin, that binds with new protein and actually activate a protein called P27 that actually inhibit cell cycle. Another medication that actually prescribed for HAR2 breast cancer patients, that is called tamoxifen. This actually inhibit a receptor called estrogen receptor that actually regulate this HAR2 protein. Okay, so this is very important, HAR2 for cancer. And the name ERB actually come from a virus called erythroblastosis virus. Okay, so this is the case of the RTK pathway, receptor tyrosine kinase pathway. They get, or actually when they bind with the ligand, they form dimer and they autophosphorate each other. Then actually phosphorate the C-terminal region of the C-terminal domain of the receptor, and then different adaptor proteins can bind. It could be GRB2, it could be PI3 kinase, it could be PLC gamma, it could be RAS gap. So by this way it actually control the signaling pathway. Now I will show you the negative feedback, because in my all signaling pathway I have mentioned how the signaling pathway can be deactivated, because once a signaling pathway gets activated, that signaling pathway should be deactivated also. And there are 4 ways by which this pathway can be deactivated. The first is like the JAK-STAT pathway called phosphoprotein phosphatase. phosphatase. I have mentioned that in case of RTK pathway, receptor tyrosine kinase, phosphorylation occur in the tyrosine domain, particular tyrosine domain. So if the phosphatase can cleave the phosphate domain, then the active signaling pathway can actually demolish. So the example like SHP1, there are many different phosphatases can present. Second number is SOCS, that also actually governed by the JAK-STAT pathway. SOCS means suppressor of cytokine signaling. I have mentioned this is actually E3 ubiquitin ligase that can polyubiquitinates the receptor and also the JAK protein. This is also function in RTK pathway. The third way by which RTK pathway can be deactivated, that is called receptor mediated endocytosis. In this case, the receptor actually get internalized inside the cell by clathrin mediated vesicles, and it actually the receptor reach to the endosome, and after few minutes, let's suppose in case of when there is no ligand, then the receptor actually come back to the plasma membrane after 30 minutes. So from the endosome, the receptor then reach to the plasma membrane. Actually cycle between endosome and plasma membrane, the cycle. So this is another way of inhibitions of this receptor tyrosine kinase pathway, but here is a difference in a particular growth factor that is called NGF. In case of NGF, that means nerve growth factor, the receptor mediated endocytosis do not actually inhibit the process, but it also actually activate the process, in which the axons, let's suppose this is the cell body of the nerve, and this is the axons, and this is the axon terminus. In this case, the nerve growth factor enter or bind with the ligand, or actually the receptor, NGF receptor, they get internalized inside the endosome. Okay, the receptor called TRK1, TRKA. This is the receptor for the NGF. They reach to the endosome, but from the endosome they can actually send the signal to the cell body. Let's suppose here the nerve growth factor or receptor are endocytosed in the endosome, they can send the signal throughout the axon and reach the cell body. This is exception. Another is fourth way is called lysosomal degradation. This is different. Lysosomal degradation. How it occurs? There is a E3 ubiquitin ligase present, like SOCS. There is a E3 ubiquitin ligase present called CCBL. CCBL. This ubiquitin ligase recruit ubiquitin conjugating enzyme, because this ubiquitin ligase, E3 ubiquitin ligase, has a particular region called ring finger, RING, ring finger domain. This ring finger domain can recruit ubiquitin conjugating enzyme, and this ubiquitin conjugating enzyme actually, let's suppose this is the receptor, let's suppose this is the receptor, then this CCBL actually ubiquitinates this receptor, and monoubiquitinations, here monoubiquitination occur, polyubiquitination do not occur. Here polyubiquitination occur, SOCS, but in this case monoubiquitination occur, and that monoubiquitination actually helps as a tag. It acts as a tag that actually helps in the transfer of this receptor into the multivesicular body of lysosome for the degradation. So lysosomal degradation actually degrade the pathway. So this is all about RTK, receptor tyrosine kinase pathway. In my next class I will talk about RAS MAP kinase pathway, because this is actually the RAS MAP kinase is very actually connected with this RTK pathway, but I will take a separate class for RAS MAP kinase pathway. So hope this video will help you. Thank you for watching, and always keep learning.